Okay.
Thank you.
Well, thanks, Jesse, and thank you for the invitation to share some of the results from the previous grape and wine cluster and the activity that I've been working on here in Vineland in the Niagara region on trying to identify an insect vector or vectors of grapevine red blotch virus, GRVV. So I'll go over this slide a little bit quickly. I think probably Jose will talk a little bit more about GRVV in the next talk, but it's become a common virus found in Nova Scotia, BC, Ontario, and elsewhere. So, foliar symptoms look a little different on red-berried versus white-berried cultivars, and there's really a progression of symptoms throughout the season. So it's certainly easier to find symptomatic plants as the season progresses. So there's also fruit symptoms, which are most problematic with changing yield quality and quantity. And up to 50% yield reductions have been reported in, I think, some vineyards in California. But really the question that we want to look at is, so we know there is some long distance spread of GRVV through infected planting material, unfortunately, but is there also some secondary local spread happening? Some data here from Wendy McFadden-Smith, who works for the province of Ontario. She and her team looked at a couple different blocks from 2018 to 2020 and sampled, I think, every vine for GRVV over those 3 years. And you can see in the Chardonnay block, there's a fairly low incidence, and the percent increase is relatively low year on year, whereas in the Vidal block, there's a much higher initial infestation in 2018, and the rate of increase is a little bit greater, and over 50% of the vines are positive by 2020. And so, again, Jose will probably touch on this next, but in BC, there's been a fairly low rate of spread observed, and roving programs have been helpful. In other places outside of California, at least a number of years ago, there was basically a lack of spread reported in New York vineyards. And over in Europe, there's been very low incidence of GRVV and no documentation of spread as of yet. So if there are going to be insect vectors of this virus, they are likely going to be in the insect order Hemiptera, which are the true bugs. GRVV is a Gemini virus, and all other Gemini viruses we know are transmitted by these bugs. And looking a little closer at the literature, it's probably transmitted by a group of bugs called the Acanorenca, which are the leaf hoppers, plant hoppers, and tree hoppers. And then the work that's been done has really focused on one species of tree hopper, the three-cornered alfalfa tree hopper, and that's found primarily in California with the scientific name Spicistolus festinus. And so it is established as the only vector of GRVV at the moment. But the alfalfa tree hopper S. festins is not established in Ontario, nor anywhere else in Canada, as far as I know, even though there has been a few records of it being in Ontario, but there are no populations. So we started with a sticky card survey of vineyards in the Niagara region back in 2019 and continued some of that in 2020. We put out a lot of sticky cards in different vineyards and different places in the vineyard. So we had some on the edges and the interiors, and we checked them weekly, basically from July to September. And we collected quite a menagerie of different hoppers, a few of them pictured here. We tested quite a few of them. And unfortunately, most of them were, at least for the purpose of research, unfortunately, most of them were negative for GRVV, but the one that stood out is this plant hopper. So the genus is Melanoliaris, and we're pretty sure the species is Aridus, but they are very hard to identify the species. And unfortunately, there's no common name, so I'll be calling it Melanoliaris throughout the presentation. And in terms of numbers, we collected 19 in 2019, and 11 were positive for GRVV, and then 24 out of 49 the following year. So that's around the 50% mark, which would be, a high enough acquisition rate that we could consider them potential insect vectors. And then I'll just point out Stictocephala here, which Deb already mentioned. In Ontario, we think the species that is most common is Bisonia, and it is a tree hopper, so it's fairly closely related to the alfalfa tree hopper, which is the vector. But in vineyards, it's very uncommon. So you can see in 2019, we only captured 2 on our sticky cards. Neither was positive, and in 2020, basically the same story. So with that data, we also looked at a couple other questions for Melanoliaris. Does the positive rate in these insects vary throughout the season? And in that graph, we're showing the total captures in blue and the percent positive in orange. So really, there's not that much variability. It's around 50% most of the time. Except you can see we had a peak in captures there in the early period where the percent positive didn't go up, so it's a little bit lower. But for the most part, I would say the answer to that question is no. We also, does the GRBV positive rate in this insect vary depending on where they were captured? And so this is the 2020 data. So you can see the total numbers captured tend to be a little higher on the edges, both in the early and the late, and the percent positives are a little lower than in the interior. So perhaps there's a trend of insects coming into vineyards from the edges, perhaps. And if they get to the interior and get captured there, they've had a longer time to feed on vines to acquire the virus. But then looking back at the 2019 data, all the insects all the Melanoliaris were captured more in this later period, but the trend is somewhat opposite to 2020. So maybe the original hypothesis is not necessarily true. Okay, and then in 2021, we looked quite a bit around vineyards. We didn't do sticky card trapping in the vineyards, but we did a lot of sweep netting in the edge vegetation outside of 9 different vineyards, just to see how abundant these 2 main potential vectors were. You can see the buffalo tree hopper is, we start capturing it in the first week of July, and there's a peak towards the middle of August, and then the numbers drop off quite sharply. And it was present in vineyard, around all vineyards. So the bottom graph is showing the 9 different vineyards, and you can see there's buffalo tree hopper at each place. Some vineyards a little more than others. In total, we captured 385, and we started testing them all for GRBV. And once we got to about 100, we didn't find any positive, so we stopped the testing. It's probably unlikely that any of them are positive for GRBV if they're captured outside the vineyard. With Melanoliaris, there were a lot fewer compared to buffalo tree hopper captured around the vineyards. You can kind of see maybe there's 2 population peaks similar to what we found on the sticky cards in previous years, a July peak and then a little bit of a peak in August. And they're not nearly as common around vineyards as buffalo tree hopper. And we only found them at 5 of the 9 vineyards. So in 4 of the vineyards, we didn't find any at all. So moving on, we could determine that Melanoliaris seems to be the only insect that is acquiring GRBV out in vineyards or near vineyards. But what about transmission? So some of the early work that was done for transmission of GRBV by the alfalfa tree hopper used greenhouse assays, and they put the insects for 48 hours on a GRBV positive vine to acquire the virus, and then 48 hours on a GRBV negative vine to inoculate it. And the results were about 20% of those negative vines became positive. So we started a transmission experiment, basically trying to replicate that method. We're not able to rear any of these insects, so we had to go out and collect them from around vineyards with sweep nets. Then we had a number of GRBV positive potted vines. We placed them in a cage with a number of periods for 48 hours with a number of insects. And then, the insects were placed with the GRBV positive vine. Then we moved the insects to another cage with the negative vine for another 48 hours. And then, of course, we killed all the insects, tested them for GRBV, and then tested those inoculated vines at different time periods after the end of the experiment. So the results with buffalo tree hopper was that we didn't find any of the vines, which we tested at 5 and 14 months after, positive for GRBV. And very few of the tree hoppers that we used actually were positive either. So just 4 out of 70. So it doesn't seem that there was any acquisition or inoculation by buffalo tree hopper. With Melanolaris, it's a little bit different. We didn't find that they were able to transmit the virus at all. So none of those vines were positive, but a larger percentage of the adults were positive for GRVV after that acquisition period. So that somewhat agrees with the field results, where we had about 50% of the individuals positive. When you cage them on vines, we got up to 74% positive. And then the work has been developing in the US on alfalfa tree hopper. So what's been found more recently is that GRVV is a circulative type of virus in the alfalfa tree hopper. That means the insect has to feed on an infective plant. The virus has to be in the gut of the insect. Then it has to be transmitted to the hemolymph, which is like the blood of the insect. And then finally, it has to get back to the salivary glands so that when the insect feeds on another plant, secretes the saliva, then the virus is injected into the phloem. So in some studies at Cornell, it looks like they suggest at least a minimum of 10 days for acquisition in order to detect GRVV in the salivary glands of the alfalfa tree hopper. So their transmission assay is now to feed or have a 15-day acquisition period, so much longer than the 48 hours used originally, then a short gut clearing period on alfalfa, and then to use excised grape leaves for a 4-day inoculation period. And even with this different assay, only 10% or 4 out of about 40 leaves were still positive for GRVV. So in 2022, we adapted our transmission assay conditions and tried to replicate something more similar to the recent results with alfalfa tree hopper. So again, we collected buffalo tree hopper from around vineyards. This time we placed them just in these smaller sleeves on infected grape plants so that they would be confined in a more tight space and hopefully feed more readily on the infected leaves. And we used a 12 to 14-day acquisition period. We had our 2-day gut clearing period on alfalfa, and then we went to transmission to an excised leaf from a negative plant for 4 days. And then after that, we froze and stored both the petiole from that leaf and the insects. And this time we decided to chop off the heads from all the insects because we wanted to test the bodies separately from the salivary glands. And the salivary glands are in the head, of course. One of the issues with this assay is that we had a really low survival of our buffalo tree hoppers from the start right till the end. So we didn't end up with many to test at the end. So this is probably due to the fact that maybe buffalo tree hopper doesn't feed very readily on grapes. Grapes is not necessarily a preferred host. The other thing is that we captured a lot of our insects in August and early September, which is kind of at the end of their life cycle. They're probably older adults. It would be better if we collected earlier in the season. But I just want to show one image of a gel here with 5 buffalo tree hoppers from one replicate where we did get some interesting results. So basically the 2 different gels just show 2 different primers for GRVV. And on each, the numbers that have the green boxes show the bodies and the other ones are the heads. And where I put the blue arrow, that's where we expect the bands if it was GRVV positive. So you can see that with both primers, all the bodies in this replicate were positive, but none of the heads were positive. And that leaf that all of these 5 buffalo tree hoppers were on, there was evidence of girdling on that petiole, and there's an example of alfalfa tree hopper girdling in that picture. But when we tested that leaf, we did not get a positive. It was still negative. And that's probably due to the fact that all the heads were still negative. The virus had not made it to the salivary glands yet. So just to wrap up, take-home message is that buffalo tree hopper is still a potential vector. It's really seems to be quite uncommon in our vineyards, but really common around the vineyards. So far, none of any of the individuals we've ever collected from the field have been positive for GRVV. But after that longer acquisition period in greenhouse conditions, we are getting higher rates of positive GRVV. But can it transmit the virus? With Melanolieris, the plant hopper seems to be a little more equally distributed in and around vineyards compared to the buffalo tree hopper. Acquisition rates in the field in vineyards are around 50%. And in the greenhouse conditions, and that in 2021, when we did that first experiment, we did get a fairly high acquisition rates in 2 days. So there seems to be an ability to acquire GRVV, but we're not quite sure about transmission yet. And so just going back to alfalfa tree hopper, a lot of the recent conclusions is that this species is still, even though it's considered a vector, it's often considered a somewhat inefficient vector. It doesn't transmit GRVV at high rates. It's it's a slow process. It takes a lot of time from feeding to transmission, but it still has epidemiological importance in California vineyards, basically meaning that it's still responsible for transmitting the virus and is an insect that needs to be managed to help manage GRVV. So with that, I'd like to thank all of you for being here today, and thanks to a lot of different people and organizations for funding, and looking forward to some questions and the discussion period.